Hello. A very good afternoon to all of you. Julia, are you able to listen to me? My voice is audible to you? Kindly let me know. Okay, because there's a fan just above me, so that's why I'm asking. Is there any type of noise or something? Then I will switch off the fan. Yes, Mansi is here. Mansi, good afternoon. Good afternoon. Good afternoon to all. So my voice is clear to all of you. Is it audible to all of you, or is there any type of noise? Because I have a fan. Okay, everything is fine. Okay, good. So how are you all, and how is your day today? Today is Sunday, so what you did today? How is your day today? Kindly let me know how is your day today. Have you done your have you ate your food? Yes, I am also good. Lots of work has to done on Sunday, so I don't know when it becomes 2 p.m. and I have to there for the class. No rest on Sunday also. So it's okay, but we have to do all the things now. So we have Mansi here, we have Nikhil here, Anushka, Juveria, Mansi, Priyanka, and Pavitra. So you all are good. I am hoping so. Just waiting for the students. Let them join also. Then we will start our session. Today we have a long session. Waiting for a positive response from your side. We will take the break in between, like for 10 minutes or 5 or 10 minutes. We will take the break because I don't want to make it complicated. So we have to do 2-3 things today. One is microscopy, one is molecular biology techniques, and then recombinant technology. So I want to know whether you have done recombinant technology in your college session or not. Whether you have done recombinant technology. Have you done recombinant technology? Jagriti, good afternoon. Like some part in the molecular technique we already covered before, but the recombinant technology is also in your this 10-day series. So I want to know whether you have done it before also. Do you know something about recombinant technology or not? I'm not getting your replies basically, maybe because of the connection issue. So just wait for your answer. So are you getting my voice? I am audible to all of you. Is there any type of noise or a hindrance in between? Then kindly let me know. Rituja is here. Rituja, good afternoon. So good afternoon to all of you. First of all, good afternoon to all of you. And I want to thank all of you for this afternoon session. So let's start it, just waiting for the students to join. And how's your day today? What do you have do today? And have you taken your lunch? Is everything is fine on your side? Yes, good afternoon to all of you. Good afternoon, a very good afternoon. So yesterday topic is flow cytometry. Are you able to get the all points which are the important one? Like what is the principle of flow cytometry and the main focus on which topics? Like we have to count the cell number, counting the cells, then to know the size of the cell, whether it's a small one or a large one. Then another is the complexity or the granularity of the cell. The cell is how much complex it is inside. Then we have the fluorescence technique to know the type of cells. We will give different type of color to different types of cells, and then we are able to recognize it on a graph. So is it clear to all of you, or is there any type of problem, or you are not able to get anything? Then kindly let me know, because flow cytometry is a very recent technique, and you must use it in your research purpose when you will do it in the future. So kindly let me know whether you have get all the basic points or the basic things which are there in flow cytometry. There are lots of other modifications of flow cytometry, or there are lots of other applications of the flow cytometry. But my main motive is to tell you the basic principle or the point on which the flow cytometry is based. One minute. So kindly let me know whether you get all the things about flow cytometry, about forward scattering, and then we have a side scattering. So kindly let me know. So how are you all? Are you getting my voice? Is it audible to all of you? Yes, we have Mansi here, we have Jagruti, Rituja, Floris, Jeet, Gayatri, and Rahul is also here. So a very good afternoon to all of you. I am asking, are you able to understand the yesterday session, which is flow cytometry? Are you able to get the points which are there, the main basic principle or the main basic points which are there in flow cytometry? Okay, you are getting it. Okay, very good, very good, very good. So now I am also asking you that how's your day today? What you have did up to 2 p.m. Is there any type of assignments or any type of work which you have to do, or are you free from the morning? What type of activities you have done in the morning? Good afternoon to all of you. Good afternoon, Lekha. So what type of activities which you perform in the morning? Devashi, good afternoon. We have our college in morning on Sunday. No, I'm not asking your daily routine. I'm just asking on Sunday basically. That is on today. So what's your morning activity? What you have done in the morning? Are you free? Are you all are free, or you have some type of assignments or work to do? So kindly let me know. Yes. No issues, no need to say sorry. I am just asking that what you have did today, or are you free, or are you doing some type of assignments or some type of college work? We have lots of activities to do. We are just waiting for the students to join. That's why I am asking that what you did today. Or are you free? You are enjoying the Sunday. Yes, yes, I know. I have to ask you another thing which is about review paper. So, Floris is here. He is asking, ma'am, kindly ask confirm about review paper. Yes, yes. So, kindly let me know. Have you think about any topic or any topic on which you write a review paper? We will start with Floris first. Floris, have you thought about or think about any topic on which you write a review paper? Then please let me know. Then we will discuss it further. So Gayatri is saying completing journals and notes. Okay, very good, very good, very good. So it's a very good thing that you are performing your daily activities or you are focused on your study. It's a very important thing basically. That's why I am asking the question only. So kindly let me know, have you think of any type of topic on which you want to work? Any topic in which you want to write a review paper, and even I ask you if you want to do it in a group or you have 2 or 3 friends which want to do it in collaboration, then please kindly let me know about it also. Please kindly let me know. Okay. Okay, I will confirm it once again from Vatsal sir. I will. It is up to basically up to you. If you are interested to write a review paper, then you must. Otherwise, no issues, no worries there, because you are said it is specifically for the internship students. So if it is only for those students and you are not interested to write it, so I am not forcing you or I am not asking you to do it. No, it's up to you only. But if you try, then it will be very useful to you in your nearby future. Devashish, I am not getting your point. Basically, you have topics. So you have topics. Then is there any type of time issue? What you are asking? Kindly clarify it to me, please. You have some topics in your mind, and then what's the problem? Okay, Hitar is saying yes ma'am, Debashish is right. Okay, okay, I will confirm it once again from Vatsal sir. No issues, not need to worry. Okay, okay. No, it's okay. It's definitely okay if you have some type of work to do. No issues at all. So if you are not interested, it's okay. It's not a forcefully job that I have to do it to you by force. No, it's only your own wish. So if you are uncomfortable or you have some, yes. So if you don't have a time or if you have some another assignments or some other type of work, then it's okay, no issues. If you don't want to do, then it's okay. Not to worry about it. So you have not to worry about it. If you have some other type of work to do, if you have some another assignments or you have your exams or it is not a requirement for you, then please leave it. No issues at all. But I am just saying, like yesterday I told you that it will be helpful for you in the nearby future. But it's okay. No issues at all. No, not at all. even if you collaborate with 2 or 3 or 4 students, then I don't think it will be that much difficult for you to write a review paper. If you collaborate with your friends, like 4 or 5 friends, together make a one paper very easily, because you have to make it of 3 or 4 pages only. So this is also another type of idea which I shared with you yesterday also. But till if you are not comfortable, then I will not force you at all. It's up to you only. So do you have? Yes, I understand. Okay, no issues, Gayatri, not at all, not at all. There is not any type of issues regarding it. Either Debashish or Floris or Gayatri, it's okay. I understand that you have to do lots of assignments and work, and then you have your paper ahead. So it's okay, no issues at all. Don't worry about it. So tell me, is there any other issue or other query which we have to discuss? If you have any other query or you have something else to say, kindly let me know. Yes, if you are writing a review, or if you are writing a review paper, or you are writing any type of a paper, then you must have a data which you have to collect it from the previous papers or from the journals, for which you have to take some time. Like if you want to write a topic on pandemic situation, then you have to collect whole data about pandemic or whole the situation in the year 2020. So definitely it will take your time. It will take maybe 4 to 5 days or maybe 1 week also. So that is up to you. I am not forcing you. You are just from graduation, so I am not forcing you that much. It's up to you. But if you want any type of my help, I am always there. So if you can write a paper of at least 2 or 3 pages, then I will help you to make it proper way, and then we will send it in a journal where it will get published in this year or maybe in January 2021. So it becomes your achievement in the year of 2020. But again, I am saying to all of you that it is up to you only. I will not force you at all. I understand you have lots of activities to do. You have your assignments, you have your papers, so I am not forcing you. I hope you will got my point. So if you have any other query, kindly let me know. And what about the other students? They are not even replying. What about Rahul, Jeet? Then we have Rituja. Are you comfortable in writing? Mansi, Priyanka, Juveria, kindly let me know. Are you comfortable? Because they have their own reasons not to write it. But are you comfortable to write it? Kindly let me know. Because you have to give me a draft only. I am here to make it proper for you. Then we will send it in a publication or we will send it in a journal. So kindly let me know what's your thoughts. We have Mansi here, Nikhil here, then Utuja, Rahul, Jeet. Kindly let me know, please. Please let me know. Okay. Yes, good. Yes, very good. Yes, I am understanding your situation also, so I am not forcing you, so don't worry. I have to check my WhatsApp chat or today's WhatsApp chat. I'm sorry, Devashish, I'm not getting your point. What are you saying basically? Okay. No need to worry. Why you are worrying so much? It's okay. I'm not forcing you all. Please, you have not to say me again and again. I know you have problems, you have assignments, so it's okay. It's okay. Don't worry. If you are comfortable in writing, then most welcome. If you are uncomfortable or you have some other types of works, obviously you are students, you have lots of work to do like assignments, you have exams. So if you are uncomfortable in writing, then it's really okay. Okay, so don't need to worry about it. Okay, so now Gigi is saying same reason for everyone. So now there is not any volunteer who can write a review paper. There is not any single volunteer. Is there anyone is? I think there should be some students from your batch because you all are very good students, and I am hoping that we will get one paper from your batch, at least one paper. Yes. So I think we should have at least one paper from your batch. Either it is with collaboration of 4-5 students or 2-3 students. Please kindly let me know. We have another students also. We have Rahul is here, Aditi is here, Lekha is here, Mansi is here. So is there not even a single volunteer to write a research paper or a review paper? Okay, so Deep is saying from his section, I think there is not any single volunteer. And what about you, Juveria? Kindly let us know. Kindly let me know, please. Yes. No, I understand, Clarice. I understand it's because of time issue basically. You have to spend 2 hours for this class also. You have your college classes also. Then you have your assignments. I understand. No need to worry. But okay, Aditi is saying yes, ma'am. Same issues. But I think we must have one paper from your batch. We must have, and even I am thinking of more than one, but at least if there are 2 or 3 volunteers, then please kindly let me know. We will make it together. I will help you. So please kindly ask in your group if there are 2 or 3 students who are interested, then I am there for helping them. We will together make a single review paper. I am there for helping those students who are interested, because I don't think everyone has his own thinking. So if there are any students, any 2 or 3 students, so if there are 2 or 3 students, kindly let me know. We will make a paper together. I will be there and we will be making a paper in which you have your own name, your institute name, and it will definitely achievement for all those 2 or 3 students. So kindly let me know if there any 2 or 3 volunteers. I think there should be. I am waiting for a positive response. I will help you. No need to worry about anything. So just you say yes, ma'am. We are here for you. Kindly let me know. If there are 2 or 3 students, please let me know. Jeet, it's your duty. Kindly confirm me within 1 or 2 days. If there, take your time. So if from your batch or from your section, if there are 2 or 3 students who are interested in writing a paper, then kindly let me know. So it's up to you, Jeet. You have to confirm me regarding it. Okay? Are you getting my point? Okay, and what about Juveria? Juveria, are you there? Kindly let me know. What are the students in your batch are saying about it? So if you are here, kindly let us know, please. Okay, take your time, 1 or 2 days, then tell me regarding it. Okay. So those students who are quite busy these days, it's okay. No issues, no worries. You have not to tell me. It's okay. It's okay. It's really okay. Okay, she messaged me on WhatsApp. Okay. 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 So she is right now she is traveling somewhere, so she will confirm later on. Okay. So we will actually I am hoping for 2-3 students, definitely for 2 or 3 students which collaborate with me, then I will help them and we will make a one review paper. Okay. Now I think that will be fine. So I will wait a positive response from your side also. So is it okay? That's all. Have you any other query? We have Floris, Jeet or Debashish. Gayatri, is it okay? So if you are busy, no issues at all. But kindly confirm from your other students or from your other friends. If they are interested, then kindly let me know. So you have the duty to ask your another friends who are not attending the session or who are not aware of this review paper. So share this thing with those friends and then kindly let me know. So today we have now everything is okay regarding the review paper. So if everything is okay, yes Devita, yes. So you can let me know later on. So now we have to move for our next topic, which is microscopy. Today we have to cover 2-3 topics. So yes, I am asking you one question. Have you done? Yes. Okay, good. So I am asking you one question, that have you done recombinant technology before or not? Have you done it in your classes or not? Because if you have not done it, then we have to start it from very basic. That's why I am asking it. Have you done recombinant technology? If not, then we will start it from the very basic. So just kindly let me know. Yes, ma'am. So you have done. Okay. So you have done recombinant technology. Actually, recombinant technology is in your this MBBT series, but then I think that you must have done it in your classes also. So that's why I am asking, because I have prepared a very basic of recombinant technology. You must know about it all, but it's okay. We have 2-3 things to do, like we have to do microscopy, then we have molecular biology techniques, in which we have PCR, we have ELISA, and then we have blotting, like southern blotting, northern blotting. So some techniques we have already done before, but some techniques are there which we have to do today. So have you done PCR? Have you know about PCR, which is polymerase chain reaction? Okay. So you have done basically all these techniques, the techniques which are your PCR or your recombinant DNA technology. So if you have known already about it, so I will cover it in today's section, because it's a very simple technique. You have known it before also. So I will not go in that deep for these topics because you are aware of these topics. But yes, we have to do microscopy first. So you all know about PCR, but these are there. These topics are there in your this MBBT lecture series. So I have to prepare the lecture on it. Like you have microscopy is there, which we have to do today. Then we have molecular biology techniques, in which we have PCR, we have ELISA, we have SDS, and all another blotting techniques which we have done previously. Then we have recombinant technology. Recombinant technology, you said you have done it. Then protein part we have done. We have done electrophoresis. We have done blotting techniques. We have done flow cytometry. And then another are recombinant applications of recombinant technology and molecular technology. So if you are aware of these technologies, I will not go that thoroughly. I will convey the message, or I will do it my best. But if you know about it, then it's very good. It will be not easy, difficult for you to get my point. So shall we start with microscopy now? First we will cover microscopy, then we will go with these PCR techniques or ELISA techniques. Okay, we will do it only fast, or you tell me how to do it. So shall we start now the microscopy? Okay, good. Okay. So today we have to go with the topic, which is microscopy. So first of all, we have to know what is a microscope, and then we will know about microscopy. So what's a microscope? It is an instrument for viewing objects that are too small to be seen by the naked or unaided eye. So basically, microscope is an instrument which is used to view the objects that are too small. So they are basically used to view the objects that are too small. Then what's a microscopy? So it is a science of investigating small objects using such an instrument is called microscopy. So when we are using the microscopes to view or to see the small objects, then it is known as, the science is known as microscopy. So the first microscope was developed in 1590. So we will get the first microscope very in the 15th century actually. Then in 1609 we have a compound microscope. So we have a compound microscope. Then in 1620 there are two persons who developed a single two-lens ocular system that was chromatically corrected. So the origin of microscope is very old. It is started from the year 1590. So these are the persons, like we have Leeuwenhoek, we have Robert Hooke. These are the scientists who worked on the microscopy, or you can say who worked on a microscope. So if you can see, this is a microscope. A microscope is an optical instrument that uses a lens or a combination of lens to produce a magnified image of an object. So with the help of microscope, what will happen? We are able to see a magnified image of a particular small object, too small to be seen with naked eye. Though the small object which we will unable to see under the naked eye, we will see it under a microscope. But how? By this magnified image of an object. So we have a magnified image of an object by which we are able to see the object which we unable to see by naked eye. So this is a microscope. We have the lenses here by which the image gets magnified and we are able to see it. So this is the basic microscope which is used nowadays. We have objective lenses, we have ocular lenses, and the objective lenses are those lenses which is placed near the object. So right here we have a slide on which we will put our specimen or our object. Then ocular lens or eyepiece are those lenses by which we are able to see the object which is unable to see by naked eye. So this is the simple microscope which are used nowadays. So these are the parts of a microscope. Like we have an earpiece. What's the function of earpiece? Which is very close to our eyes. It is to observe specimen and enlarge the image formed by the objective lens. So we have this one is the objective lens. So they may be of 10x, they may be of 40x. So there are lenses which will magnify the image of a object. So you can see eyepiece to observe specimen and enlarge the image formed by the objective lens. We have objective lens to focus and enlarge the specimen. So objective lens is the one which enlarges the specimen, or you can say which magnifies the specimen. We have knobs to adjust the focusing or for the focusing purposes. So we have knobs. Where we have knobs, like these are the knobs. By this we can put it nearby or we can put it far away. So we have knobs for adjustment purposes. Then we have a stage to store the microscope slide. So this is the stage on which we will put our microscope slide. Like here, this is the stage on which we will put our specimen or we will put our slide. So we have the stage here, and these are the knobs for focusing. So these are the points for focusing. Then we have the clips to keep the microscope slide in a position. Then we have diaphragm to control the amount of light entering the lens. Mirror is there. Then we have a condenser. Yes, this is also an important thing. We have a condenser to focus light on the specimen. If you can see here, there we have a condenser to and this is a source for a light. Like we have a light from here and it will put on the specimen. So this is a slide. From the below side we will put a light on this specimen. So in between it, that is in between this slide, that is in between this specimen and this light, we have a condenser. So we have a condenser, and what's the function of a condenser? To focus the light on the specimen. So we will condense the light. Are you getting my point? We have a condenser in between the source and the specimen, and it condenses the light on a particular specimen. So then we have lens tube. We have a lens tube to hold the lenses which magnify the specimen. Then base and limb are the another part of a microscope. But the main thing is this one. We have the knob for adjustment. We have a eyepiece, or you can say ocular lens. We have objective lens. These may be these lenses just magnify the image. We have a condenser. This condenser is present in between the light. Like this is the switch on by which light will come here and it will put on this specimen. But in between this we have a condenser which will focus the light on the specimen only. So we have a condenser also. So this is the terms which are used in microscopy. Basically we have magnification. What do you mean by magnification? It is that how much enlarged the specimen or the object becomes. So it's a degree of enlargement, like we have only that much small specimen which is unable to see by the naked eye. So magnification is what? When it becomes big in size and we are able to watch it under the compound microscope. So number of times the length, breadth and diameter of an object is multiplied. So this is known as magnification. It depends upon optical tube length, focal length and magnifying power of an eyepiece. So these are the things on which the magnification depends upon. So hello. Hello. So students, are you is it okay now? I'm sorry, what's happening? Oh, I'm sorry. I think someone just called me and my phone gets disconnected. Now I am audible. Kindly let me know, please. I am audible now. Please kindly let me know. Yes, I will start from the magnification only. Okay. So I am here. So I am telling you about the magnification. It is the degree of enlargement. So we have a small specimen like here. With the help of microscope, this object is, we are unable to see it under the Okay. I hope you will get it now. So I am telling you about this thing that we have magnification. So what is basically magnification is, like we have a small object here. And we are unable to see it by the naked eye. So with the help of a microscope, like here we have a microscope. With the help of a microscope, we are able to see the large image. So with the help of a microscope, this small object which we are unable to see by naked eyes, so we are able to see the large image of the small object which we are unable to see by the naked eye. So this is what is magnification basically. It is the degree of enlargement. That how the image gets enlarges. So number of times the length, breadth, diameter of the object is multiplied. So the diameter of the object gets multiplied, and it depends on many factors like optical tube length, focal length and magnifying power of the eyepiece. All these are the part of a microscope, like focal length of the objective lens and the magnifying power of the eyepiece. So what is basically magnification is, it is the degree of enlargement of small specimen which we are unable to see by naked eye. So the total magnification of eyepiece The total magnification is magnification of the eyepiece into the magnification of the objective lens. So this is a part of the microscope by which we get magnification or the enlargement of our small object or a small specimen which we are unable to watch by naked eye. So this is the number one term, which is magnification. Now we have number 2, which is resolution. Now what's resolution? It is the ability to reveal closely adjacent structural details as separate and distinct. So what is resolution? Basically, suppose we have nearby two small structures. So what is resolution? Basically, whether we are able to see it as a little bit without clarity. Resolution is with clarity. So when the two objects are placed nearby, we are able to see them with full clarity. So this is known as resolution. Like we have a limit of resolution, but resolution is what when we have two objects and we are clearly know the distance, or we are clearly able to see the two objects. So this is known as resolution. It is the ability to reveal closely adjacent structural details. So if two subjects or two specimens, they are placed together, like these are in like that way. Suppose it, let me, 1 minute please. Suppose these are the two subjects or two specimens. We are unable to see it by naked eyes, but when we put it under a microscope, so there is a microscope, what will happen? We are able to see it like that. So this is known as resolution. Then we have limit of resolution. The minimum distance between two visible bodies at which they can be seen as separate. So we are able to see it as a separate and not in contact with each other. So they are, if they are like that, like they are in contact with each other, but what we will see? We will see it like that way. So the minimum distance between two visible bodies at which they can be seen as separate is known as limit of resolution. And the limit of resolution is proportional to the wavelength and is directly proportional to the wavelength and inversely proportional to the numerical aperture. So this is resolution. Then we have numerical aperture. Let me check whether you are getting it. So are you getting my point? We have magnification. What is basically magnification? It is the enlargement of a small object which we are unable to see by the naked eye. And then the another thing is resolution. What is the resolution? It is the ability to see the two objects which are nearly placed as different ones. So are you getting my point? Kindly let me know. Is it buffering now also? So are you understand what is magnification and limit of resolution? Kindly let me know, then we will move further. Kindly reply please. Is it okay now? So that we will move further. Okay, and I'm sorry Devashish, are you getting it or not? I am not understanding what are you saying. Are you getting my point? If there is any doubt, kindly tell me out. Kindly tell me now. So are you understand magnification and resolution? Or is the signal is okay now? First of all tell me this thing. Is it okay now? Then we will move forward. Kindly reply please. Okay. So shall we move further? So Debashi, is it okay to you? Okay. I'm sorry. Because of buffering there is a problem. Sorry for it also. Okay. So now we will move for the next. So we have two things. One is the magnification, which is the enlargement of the specimen or the object. Then we have a resolution, which is the ability to separate the closely placed objects or closely placed specimens which can be separated and not in contact with each other. So we are able to separate them and known as limit of resolution. This limit of resolution is directly proportional to the wavelength and inversely proportional to the numerical aperture. So what is numerical aperture now? It is the ratio of diameter of lens to its focal length. So numerical aperture is basically what, if you see here, ratio of, it's a ratio, first of all it's a ratio diameter of a lens to its focal length. So we have a diameter, like diameter of a lens to its focal length. So this is known as numerical aperture. So it is the capacity of an object. Basically what numerical aperture is, it's the capacity of an object to render outline of the image of an object clear and distinct. So with the help of numerical aperture, we are able to see the object clear and distinct. This is also a part of a objective lens or the focal length. These are all the part of a objective lens basically. Then this is also the distance between front surface of a lens and the surface of cover glass. So what they are saying here, we have a slide. This is a slide on which we have our specimens. Suppose here we have a bacteria. Down here we have a light here, and in between the specimen and the light we have a condenser lens. So right here we have a condenser lens. So you can see here, specimen is here and the lens is placed here. Specimen is here, lens is here. Suppose this is your lens, objective lens, and the specimen is here. So the angle or the angle which is made is known as working distance. Like here the distance is more, but in this diagram the lens is very near to the specimen. Suppose specimen is here, so the angle is different. So this is known as the distance between the lens and the specimen. So our main focus is on objective lens basically. So it is the distance between the objective lens and the specimen which is present on the slide. Then another term which is used in case of microscopy is contrast. It's the difference in intensity between two objects or between an object and background. Contrast is basically what happened basically, we stain the specimens. Some specimens are, you can say, transparent. So we are unable to see the transparent specimens. So what we do, we just give the stain to them. So when we give them the stain, so what happens, it is a difference in intensity between two objects or between object and background. So when we stain it, so there becomes a contrast between the specimen and the background. So this is known as contrast. This thing will come when we will do the phase contrast microscopy. What happens in phase contrast microscopy? We stain the transparent specimen so that we will be able to see it properly. But only the specimen gets stained and the background is either it is transparent or of any color. So it is known as contrast. It is between the object, that is the specimen which we stain, and the background. So this is known as contrast. Contrast, we all know, like contrast is the one color and the other color. The contrast between the two colors, like we say if we have to make any type of costume also, we check their contrast, whether they are matching with each other or not. Like that thing happens now. So contrast is between the two different colors. So then these are the properties of some microscope objectives. Like we have magnification power. According to these properties, according to magnification, according to resolving power, or according to their refractive index, we have different type of microscope. So now there are very advanced microscopes are there. So these are the properties like magnification, numerical aperture, focal length, working distance. Working distance is what basically the specimen and the objective lens. The distance between the specimen and the objective lens is known as working distance, and the resolving power. So we have different type of microscope nowadays which are based on these parameters. When we have the simple microscope, the magnification is very less, the resolution power is very less, so everything is very low. But now we have advanced microscope on which the magnification is like here you can see the magnification is 40 to 45x. So this is very primitive microscope where the magnification is very low. But we have now the microscopes, or you can say we have now the lenses whose magnification power is very high. Like here we have 40 to 45x. And then we use oil immersion also. Oil immersion is a technique in which we put the oil in a specimen, the magnification will become 100x. So if the size of the specimen is suppose x, with the help of this magnification it becomes 100x. So the size becomes that much enlarged with the help of oil immersion. So x specimen will become 100x and we are able to see it under a microscope. So these are the points. Like we have, these are the parameters basically on which the microscopy or the microscopy or the microscope techniques work nowadays. So we have very advanced microscope nowadays. So then we have numerical aperture, then we have focal length, we have working distance. So if you can see here, the distance in the simple microscope is up to 20 mm. But in the recent microscope, the distance becomes very less. Like here you can see, this is the primitive type of microscope, but this is the advanced microscope. The distance becomes very less. So these are the parameters on which the microscopy field is generally based on. So magnification, numerical aperture, resolution, refractive index, these are all the terms. Then this is some of the physics part. There is a refraction. What is refraction? If the light enters the glass at any angle other than the right angle, a deviation in the direction will occur. In addition to retardation is known as refraction. We all know about refraction, that when the light will put on a certain substance or a glass, it will deviate in certain direction. So it is known as refraction. So a curved lens will exhibit both refraction and retardation. All these things you have done in physics also in your 9th 10th class. So you have known about concave lens, and then we have convex lens. We have concave lens or convex lens for nearsightedness or farsightedness. You have known it before also. So what happened in this basically? This is the ray. This is the ray which will put on the substance or an object, then it will deviate in some other way, or it will change its path basically. So this is the angle of incidence. What's the angle of incidence? When the ray fall on the object, then by falling or by touching the object, it will move somewhere or it will refract back to somewhere. Then it's known as angle of refraction. So we have angle of incidence and we have angle of refraction. That when the light touches the subject or a specimen, it will refract in some other direction. So these are the terms which you know: diffraction, then we have dispersion, This is the magnification. This is the important thing which we have covered. This is the enlargement of an object. Then resolving power. Resolving power is defined as the distance separating the 2 points. So we are unable to see it under naked eye. So this is the resolving power, that the distance separating 2 point objects within the specimen that can just be distinguished from one another in a image. So this is known as the resolving power. I will generally focus on those points which are important in microscopy. So this is, if you can see here, this is object, this is the specimen, and these are all we all are here. So you can see here, and this is the lens, this one is the lens. So basically the object is present here. When before the object we have light also, we have convergent lens also. What will happen when we see it under this lens or a converging lens? Converging, the whole lights will get collect in a single point. So these all images, or you can say these all light rays, they collect it on a single point, and there we found the image. So our eye will see this image. So this is the object. The object is of this shape, but when we see it from here, it becomes like this shape. If you can see here, the size of the object is small, but when we see it under a lens, what will happen? The size of the object will become larger in size. So the size becomes larger with the help of this objective lens, or you can say with the help of this converging lens, we are able to see this small object into the larger one. So this is a basic criteria or the basic principle of microscopy, that we are able to see the small object into the larger one. So these are the types of lenses which are basically used. Like this is another type of lenses, and these are only the rays. So they are just saying that how the rays will work. There are some type of abbreviations also, like some type of defects are also there in case of microscopy. So this is in the simple microscope which you have seen in your school days, or I hope you must perform some experiments also. So we have a convex lens which acts as a objective lens. Now this convex lens, what it do? This is the object which is present, which is small, and when here we are present, when we see it, it is larger in size. So a light ray parallel to the optical axis of the lens passes through the focus of the lens, while a ray passing through the center of the lens does not bend. Now here two things happen. Here is the one ray which passes through the center of the lens, it will not converge. Only that ray will converge which will passes to the side by. So you are getting my point. So this is the object. Suppose 1 minute. So suppose we have an object here. Suppose those lights, we have a object here, and this is a lens. So those rays which pass through the center of the lens, they will pass as such. But there are some other rays which passes from the sides. Those rays will converge. So these rays which passes from the center, they will pass as such, and those rays which strike the another area of the lens, they will converge, and they will form the magnified image which is quite larger than the actual object. So this image is quite larger than the actual object. So this is the basic principle of the microscopy. So this, if you understand this point, then you will get everything, because this is the objective lens, also the convex lens, which will magnify the image of the given object. So are you able to get it? Let me check first, is everything is okay? So are you getting it? Yes, you are getting it. Now, is there any problem in the is there any buffering now also? Yes ma'am. Is there buffering also now? Yes. Oh. But the signal is good here from my side. Are you getting my point? Is there any buffering now? Is there any buffering now? No. Okay. Thank you all. So let's move me further. Before it, I will show you one simple microscope video. 1 minute, please. So this is a very simple microscope which I want to share with you. They are also buffered. Okay, let them take its time. I will show you the PPT then.
An activity. Built on trust. View larger than life. A practical introduction with projects and activities. Microscopes, parts and their uses. A. The eyepiece, ocular, is the part of the microscope closest to your eyes, through which you look. It contains the ocular lens, which makes the image produced by the objective's lenses larger. An eyepiece may be labeled with its magnification or power, for example, 5x or 10x, meaning it enlarges 5 times or 10 times. B. The body tube holds the eyepiece in position over the revolving nosepiece and the objectives. The body tube may be straight up and down or slanted. C. The arm is the curved metal piece that holds the body tube in place over the stage and the base. D. The revolving nosepiece holds the objectives and allows you to change objectives while looking at a slide or sample. E. The objectives are the parts at the bottom of the body tube closest to the sample you are examining. Each objective has a lens and a tube-like holder, the mount. The longer mounts hold the stronger lenses, the high-power lenses. The shorter mounts hold the weaker lenses, the low-power lenses. Like the eyepieces, objectives also come in various parts. Microscope have several objectives, for example, 4x or 5x, 40x or 45x, and 100x. Some microscopes only have 1 or 2 objectives. F. The stage is the flat surface on which you put your slides or samples. G. The stage clips holds the slide in plate on the stage. H. Beneath the stage in all the besto microscope have a condenser having standard diameter of 39.5 mm, which collects and concentrates the light before it passes through the sample. I. The iris diaphragm is used to control the diameter of the cone angle of the light transmitted onto the specimen. J. The coarse adjustment knob is the large knob used to adjust the position of the body tube, allowing you to quickly bring your sample slide into view. K. The fine adjustment knob is the small knob used to change the position of the body tube by making small adjustments to the focus of your sample. Almost all of our besto microscopes have both coarse and fine adjustment knobs, but some have only one knob. L. The mirror, located beneath the stage diaphragm, increases the amount of light shining through your sample or slide. Sometimes we use lamp plugged into an electrical outlet instead of mirror or reflector. M. The base of the microscope is the heavy bottom part. It supports all the other parts of the microscope. N. Substage adjustment knob is used to move condenser up and down, bringing it into focus. You're probably anxious to get started using your microscope. Before you use your microscope, you need to know how to set it up properly. This video contains instruction for you to follow. You should come back to here each time to use your microscope until you know the instructions by heart as well as the microscope. Step 1. Remove the besto microscope from its box or cover. Always pick up your microscope correctly. Grasp it firmly with 2 hands, one hand under the base and another on the arm. Place the microscope on a table, away from the edge. Move it to a position in which you can look comfortably through the eyepiece. You may want to sit on a chair or stool. Step 2. Make sure low power objective is in place over the hole in the stage. Use the coarse adjustment knob to raise the objective so that its lowest bend is about 1 inch, 2.5 cm above the stage. In all besto microscopes, aperture control is by iris diaphragm. At this stage, open it fully. Step 3. Put a slide on the stage so that the center, the bit you want to look as, is over the hole and under the lens. Use the stage clips to hold the slide in place. Step 4. Look through the microscope. You should see a circle of light against a black background. Twist the plano concave mirror around to make the circle as bright and even as possible. If you cannot see a circle of light, your objective lens is probably not in line with the ocular tube. Adjust the nose piece until you hear the lens click into place. Step 5. The reflector has one side flat, while the other is concave. It is difficult to say which one is best to use in every case. You will have to experiment to see which gives you a better view. The flat side will provide a sharper image in most cases, while the curved side will provide a brighter image. Here's a helpful hint for you. Never use your microscope in direct sunlight, as the reflected light could damage your eyes. Step 6. If you are using a lamp of your lighting, put it about 20 cm away from the microscope. Adjust the lamp so that the light points towards the mirror under the stage. Step 7. We have already told you in Step 2 that low power objectives is in place over the hole in the stage. Look through the eyepiece and use the coarse adjustment knob to focus upwards or move away from the sample or slide. This should bring the slide or sample into view. If you go too far, simply begin again at Step 2. Step 8. When the specimen slide comes into focus, it will become sharp and clear. Step 9. To use the high power objectives, turn the revolving nose piece to bring it into positions over your samples or slide. You should be able to see the sample or slide through the high power objective, so you should only have to adjust the fine adjustment knob. Step 10. Now adjust the aperture control with the help of diaphragm. Close it until circle of light dims and then open it again slightly and stop it where you get maximum resolution of slide or sample in view. Step 11. Medical microscope model Besto 10 have a special type of lens called an oil immersion lens 100x. This lens is used by placing a drop of oil, cedarwood oil, on the top of the cover slip and adjusting the position of the front lens of the objective so that it is touching the oil drop. The oil bends the light more than the air wound, so more light can enter the microscope. Oil immersion lenses make it easier to see samples because they increase the amount of light going through the sample or slide and microscope and into your eye. Convert your simple microscope into LED illumination microscope with LED light source. Our Besto LED light source eliminators the variation that due to reflector light or hologram lamp technologies. Features: some interesting thought entire lifetime. No lamp replacement required, no extra cost. No lamp revalidation required, no wasting time. Lower power consumption and environmentally friendly by workable on 2 pencil cells of 1.5 volts. Rubber feet. Microscope with rubber feet prevents skidding and scratches on the work table and also provides positive grip.
So that's all for a simple microscope which you must see it in your schools or in your college also. So it's just a simple microscope which we have seen just now in our video. But now we have lots of other advanced microscopes. The techniques are now very advanced. So now we will go further on the PPT. So this is the thing which we have done. That this is the main point of any type of microscope, whether it is a simple microscope or an advanced microscope. So you have the object of a small image and you will be able to see the magnified image. This is the first principle on which the microscopy works. And what happens here, like we have a convex lens which acts as an objective lens. So this is objective lens. So the rays which pass through the lens as such from the center, they will not bend, so they will pass as such. But other rays which are, like they are saying here, a light ray parallel to the optical axis of the lens passes through the focus of the lens, while a ray passing through the center of the lens does not bend. So this ray does not bend, and the other one which passes parallel through the lens, they will bend and they will form a magnified image. So this is the basic principle of any type of lens. You have to remember this thing only, that you will get a magnified image of a specimen. Then after it we have key parts of the microscope. The main important part in the case of microscope is basically the objective lens. So this is the objective lens. The objective must gather light coming from each of the various parts or points of the specimen. So if you can see here, from this is a specimen and you will get the light from each and every part. The objective must have the capacity to reconstitute the light coming from the various points of the specimen. So we are collecting the rays. You can see here this is a specimen, but below it we have a lamp. So light came here and it will deflect in all these directions. From this light which will put on a specimen, the light will deflect in all the other directions. So the objective must have the capacity to reconstitute the light coming from the various points of the specimens into the various corresponding points in the image. Then this objective lens will collect all these rays into a single point. Now if you are able to see, here the specimen is that much small, but here the specimen is large one. This is small black dot and this is a little bit large black dot. So the objective must be constructed so that it will be focused close enough to the specimen so that it will project a magnified real image up to the body tube. So this objective lens is very much important in case of microscopy. So if you can see here, where is the microscope? If you can see here, this objective lens, where we put the specimen, from here the light will come here and the light will move in many directions. These objective lenses are very much important to get a real magnified image. We are here from eyewitness we are able to see it, but the every type of focusing is done by the objective lens. So objective lens are very much important when we are studying the microscopy or when we are working on the microscope. So this is the objective lens which will convert the rays which come from the specimen, and we will able to see the larger image. So we will see the magnified real image of the specimen. Then we have some types of microscopes, like we have simple microscope, we have compound microscope, we have many more advanced microscopes also. So number one is the light microscope, which we have seen in the video, in which we don't have any type of bulb or any type of light. We have to take it from the sunlight or from the outer environment. So we have to use it sunlight or any artificial light, or you can say you can use a bulb or any type of light source. So there are four types of microscope which we have to study in the light microscopy. We have bright field microscopes, dark field microscopes, phase contrast microscopes, and we have fluorescence microscope. So these two microscopes are very important, rest are very simple ones. Then we have electron microscope, in which we have to study transmission electron microscope and scanning electron microscope, in which we have to study SEM and TEM. So we will start with the light microscope. In the light microscope, the number one is bright field microscope. So what's happened in bright field microscope? It produces a dark image against brighter background. So what happened? The background is brighter in color, and we will get a dark image. So that's why it is known as bright field. That is, the outer environment or the outer background is brighter in color. Has several objective lenses. Two types are there: simple lens and simple microscope and compound microscope. So if you are using the sun as a source of energy, sun as a source of light, so it is quite bright. So we will getting the bright field, and it produces a dark image against the brighter background. So in simple microscope we have a single magnifying lens, that is the objective lens. We have a single lens. But in compound microscope, which is used nowadays, we have series of lenses for magnification. We have number of lenses which will magnify the particular image or which will magnify the particular specimen. So light passes through the specimen into the objective lens. Oil immersion technique is here. So we have also seen in the video that we have an oil immersion technique, in which we put the oil on a specimen and the lens is just placed above the oil, or even it touches the oil. So by which we will get the 100x magnified image. So this is the thing. Then we have one or two ocular lenses. What are the ocular lenses? Which are basically the eyepieces. And here the resolution is approximately 200 nanometers. So in the simple we have single glass, or you can say single lens, but in compound microscope we are using series of lenses. I will show you. So this is the effect of immersion of the oil, which we have seen in the video also. If you can see here, this is a specimen, and above the specimen we have a slide to cover the specimen. Otherwise, when we take the slide again and again, what will happen? The specimen may be damaged or may be washed out somewhere. So we have a slide, or we have a specimen. Above it we have a cover slip. So this is a cover slip, very thin and made up of glass basically. So what will happen? This is our lens. This is our lens. This one, and this is also our lens. Now what will happen? If you can see here, here is a distance between the specimen and the lens. We know there is a distance between the specimen and the lens. If the distance is more, the resolution or the visualization is less, which we have done in the previous slides also. Let me show you. So if you can, yes, if you can see here, if the distance is more, known as a working distance, if the distance is more, the clarity will be less. So what happen in case of, what happen here now, what happen in this case, if we add the oil, the distance also becomes less, plus the whole light source or the light rays, they will converge onto the objective lens directly. So if you remember the first point, that the objective lens will get whole light rays and converge it to make a image. So if you can see here, some rays will pass as such and they are not focused on the lens. But here when we are using the oil immersion, what will happen? All the rays will focus on a optical, or you can say objective lens. That is our main point. We have to know this thing only, that whole light sources will converge into the objective lens, then we will get a magnified image. Then we will found a magnified image. So this is oil immersion. By this we were able to get a magnified image with the help of a immersion oil. So if you can see here, this is again the basic, very basic simple structure. This is a light as a source. It may be a sun or maybe a bulb. So this is our specimen which is present here, and in between the specimen and the light we have a condenser lens which will condenses the light into the specimen. Now from the specimen, look at the distance up to the objective lens. You have to know the distance between the two. From here, the rays, these light rays will pass and they will found a image. So you can see here on the objective lens here, these rays converge together. So the light rays will converge together and then they will form a image. This is a specimen and this is a specimen. So both are the same specimens, but you have to know the magnification which occurs. The small specimen becomes a large specimen with the help of this objective lens. So our main focus is only this objective lens which helps in the magnification of the image. So this is the optics, or this is the principle of the compound microscope, that we are able to see the magnified image of a object. So these are the advantages or disadvantages of the simple or compound microscope, that these are used to view the live cells. So you are able to view amoeba, you are able to view paramecium, or you are able to see the bacteria which are present in the curd. So I think you have performed it before also, so you are aware of it. And some disadvantages are there, that they are of low contrast and need to be stained. So we are unable to see them as they are transparent, and we have to stain them to see the cells or the specimens. Even you have performed the cells from the buccal, from your mouth, from the saliva part also. You must have performed that experiment also. So these are some of the disadvantages. So these are the types of results which we see under the microscopy, in the simple and the compound microscopy. But we have to stain the specimens first. By staining you will see the specimens. I think you have performed it before also. Then we have another microscopy which is known as dark field microscopy. Now what happens here, it produces a bright image of the object against a dark background. In the simple and the compound microscope, the background is bright and image is dark. If you can see here, the background is bright and the image is darker in color because of staining, or you can say. But what happened here? Here the background is dark and the image is bright. So optical system enhances the contrast of unstained bodies. So here we don't need any type of staining or any type of staining to make it darker in color. So we are able to watch the transparent specimens also. So specimens appear gleaming bright against darkest black background. So this is a general structure of a dark field microscopy. Here we have. So the only thing is that here we have background dark and the image is brighter in color. In the previous one, the background is light and the image is darker in color. So if you can see here, similarly everything is same like that one, but here what happened, the specimen is present here. So the background is dark. So we have a dark field background. This is a specimen, and when we see it under the objective lens, we are able to see the image, but the image will be brighter in color. So this is a specimen. Look at the condenser. In the bright one, the condenser is very small. Here the condenser is quite large and objective lens is small. In the previous one, if you can, in the previous one, let me show you here. The objective lens is larger in size and the condenser is smaller in size. Are you getting my point? The objective one is larger in size and this one is smaller in size. But now what happens? The condenser is larger in size and objective lens is smaller in size. In case of dark field microscopy, the condenser lens is larger in size, but the objective lens becomes smaller in size. So if you are able to see it now, the lenses are smaller. So you can see the objective lens is small and many rays are passing as such. So this is the dark field microscopy, and in it what happens, basically the background is dark. If you can see in this image, the background is dark and the specimens are shining or they are brighter in color. So they are dark field microscopy. So these are simple setup, and you don't need to use you don't need to do staining. Even you can use the unstained or the transparent specimens, because if the background is black, you can put anything white or transparent, you are able to see it. So this is the dark field and the bright field microscopy. So we have done dark field and bright field. Now we have another two microscopy which we have to do. So look at the dark field. You can see the condenser lens is larger one and the objective lens is the smaller one. This is an important point. So this is your dark field microscopy. Now we have to move to phase contrast microscopy. But before it, let me check whether you are here. So are you getting my point? Are you able to understand the things? Are you understand the difference between dark field microscopy and bright field microscopy? Because now we have to move with the phase contrast microscopy, which is quite important. Okay, good. So now we will move to the third we have to do 4 microscopy only. Simple microscopy, that is dark field, bright field, and phase contrast microscopy and fluorescence microscopy. So we have 2 microscopy more, and then after it this topic will be okay. So now we will move with the phase contrast microscopy. Jeet, I am noting your points or your queries. I will answer it. Let me complete it first, then I will answer. Okay. So you have not to worry about it. I will answer it. So we have bright field microscopy. Then we have dark field microscopy. Now we have to move further to the third one, which is phase contrast microscopy. Obviously, from the name suggests, it will be definitely a little bit advanced from the previous ones. So let's do it now. Okay, now we have another microscopy which is known as phase phase contrast microscopy. It is started in early 1934 because the microscopes which we are using nowadays, they are quite old. They are used from the 1500 century. So by 1934 we are able to get the phase contrast microscope. So they produce high contrast images. First of all, the important point that they produce high contrast. Contrast means, like we are saying, contrasting colors, contrasting suits. Contrast means two types of colors, one dark, one light, one dark. These types of shades are basically known as contrast. So what is basically why these phase contrast microscopies are used? The main advantage is living cells can be examined in their natural state. So we don't need to do any type of staining, or sometimes what happens, because the staining is quite toxic in nature. When we are doing the staining to the live specimen, what will happen? The specimen will get dyed. So the advantage of this technique is what? That living cells can be examined in their natural state. So without damaging their natural state, we are able to examine the living cells. So on what principle phase contrast microscopy works? It is an optical illumination technique in which small phase shifts in the light passing through a transparent specimen are converted into contrast changes in the image. Basically, two things which you have to keep in mind. Number one, here we are using transparent specimen without any type of stain. Okay. And number two, we will get contrasting changes. So it is an optical illumination technique in which small phase shifts in the light passing through a transparent specimen are converted into contrast changes in the image. Light rays in the face produce brighter image. Light rays out of the face form the darker image. So we will get two types of image. When they are in the face, they are brighter in color. When they are not in the face, they are darker in color. So same specimen is giving two types of color changes. So contrast is due to the phase rays. So I will show you in the next slide, but you have to remember 2 points in phase contrast microscopy. Number one, we can use transparent specimens and we don't need to do any type of staining, because when we are performing it with staining, lots of the inner structures of the living cell gets damaged. So no need of doing staining. We can use transparent specimen. Then we get two type of contrast. We get bright images as well as we get dark images of the same specimen. So these are the points which you have to keep in mind. Okay. So now let's see. So suppose this is a bacteria or this is a specimen or a living specimen. Now what will happen? Rays deviated by specimen is 1/4 waving length out of phase. So when the rays pass through it, if you can see, here we get this wave. Otherwise the waves are same, but here the wave is this one. Now deviated ray is half length out of phase. So we have this wave on this phase plate, and then we have an objective lens. Now we have to measure the wavelength from both the in phase and out phase. So if you are able to see here the shape is like that, and here the little bit difference in the wavelength. So these are the rays which pass from the specimen. These are the rays which passed as such, but they have to move along with the phase plate. So when they get joined here, both these rays, if you can see here also, the wavelength is in downward direction, but here those which do not pass the specimen, the wavelength is in the upward direction. So we will get the two types of wavelengths, you can say. Then they pass through the objective lens and we will get image here. So deviated and undeviated rays cancel each other out. So both rays combine then. So by these two waves, or you can say by these two wavelengths, we are able to generate the contrast. Let me show you. So this is a part of the microscopy, which basically we have a condenser annulus or annular diaphragm is a opaque flat black plate with a transparent annual ring, produces hollow cone of light. I will have to show you the diagram. Okay, now here. So we have condensers. Now we have the specimen here. So this is quite advanced technique. So you can see the microscope here is quite long, or you can say quite different from the simple microscope. So we have a condenser annulus. This is the condenser annulus which will be present on the lower side, and on the upper side here we have a phase plate. On the upper side we have a phase plate. This phase plate on which we will get different wavelengths. So what happens here, we have two condensers, the light will pass through it, and here is a specimen. From specimen the light will pass through two objective lenses. So we said in the simple microscope only one magnifying lens is used, and in the complex microscope is more than one type of magnifying lenses are used. So here we are using two lenses, right here and right here. So what will happen now, when they will pass from this phase plate, this phase plate, what will happen, we will get the image but with contrasting colors. Let me show you one more thing. So this is the phase contrast microscopy, in which you will see the specimen but in contrasting colors. One is brighter in color, or you can say lighter in color, and other one is the darker in color. So this is known as phase contrast microscopy. So what happens in phase contrast microscopy, you are just placing the light cell. So little difference in contrast between the cells and the water provides very high contrast as compared to bright field and dark field microscopy. So image is generated from both diffracted and undiffracted lights. So what happens here, they are saying basically this thing. One minute. So they are saying that image is generated from both diffracted and undiffracted light. So if you are able to see here, this is image is undiffracted. This will pass as such. They do not pass from the specimen. But this is a diffracted light which passes from the specimen. But the image which we got here is from both these lights, from diffracted light as well as from the undiffracted light. So this is the thing, that image is generated from both the diffracted, that means which passes through the specimen, and the undiffracted light. So if you are able to see here, we have two condensers here, we have objectives here, and then we have the phase plate. And even if you can see, the color of the light ray is light purple here and dark purple here. Let me show you again. So if you are able to see it, the color of the light ray, which is like here it is purple only, but when it passes from the specimen, you can see, like this is a specimen, some rays pass from the specimen and some rays passed as such. So you will see the diffracted light which is from here, which passes from the specimen, this light purple one. Are you getting my point? So this light purple one which passes from the specimen, they are light purple light, and those one which didn't passes from the, you can say, specimen, they are dark purple in color, and there is a face plate here from which it passed. So you have to remember one thing only, that we get a contrasting type of image here. Those rays which pass from this specimen, they are, if you are able to see, they are light purple in color, but these are known as diffracted light. But some rays or some light will passes as such without touching the specimen or without entering in the specimen. They are known as undiffracted light. So they will pass from the face plate and we will able to see the image here. But the main thing is that image is generated from both diffracted and undiffracted light. So the light which will pass as such from the center, this is the diffracted light, and one is the undiffracted light which will not touch the specimen or which will move as such. So we have two types of contrasting lights here. So generally these are used in microbial mobility, shape of living cell, detection of endospores and other functions of the body. But the basic principle is here is that we have two lights, one is diffracted light and one is undiffracted light. But the final image is found from both the diffracted as well as undiffracted light. So this final image is formed from both. If you can see, this is a face plate, so this is the diffracted light which passes from the face plate, and this is the undiffracted light. So the image is found from both diffracted as well as undiffracted lights. One more thing is here, the ray deviated by specimen is 1/4 wavelength out of the phase. So when the ray passes the specimen, the wavelength gets changed. Ray deviated by specimen is 1/4 out of the phase. So when something is like in flow cytometry we also seen yesterday, when the light touches the specimen, when it will move in forward direction, in the side direction, their whole functioning gets changed. Like forward scattering is for the size of the cell and side scattering is for the functionality or the complexity of the cell. Similarly, when the rays pass from the specimen, ray deviated by the specimen is 1/4 wavelength out of the phase. So this rays will get out of the phase. If you are able to see here also. Let me show you again. If you are able to see here, both are the same rays of same wavelength. So both are the rays of same wavelength, but here what we get? Here the image is like this one, but here the wavelength gets changed by 1/4. So this diffracted wavelength gets 1/4 out of the phase. So then again when it passes from this phase plate, it's already 1/4 out of the phase. When it passes from this phase plate, now it becomes totally changed. You can see here the wavelength is in downward direction, but in this case the wavelength is in the upward direction. So when it reaches here, deviated ray is 1 second wavelength out of phase. So half wavelength gets changed. You can see here the wavelength is in upward direction, but in the deviated ray, the wavelength is in downward direction. So the wavelength gets out of phase when it touches the specimen. So are you able to get my point? When it touches the specimen or when it gets passed from the specimen, what will happen? The thing which happen is that it gets half wavelength out of the phase. So it is in the upward direction, but here in this case you will see the wavelength gets changed, but we have to combine the result of both undeviated and deviated wavelength. So this is the phase contrast microscopy basically. Let me show you if I have some another diagram. No. So this is a phase contrast microscopy in which the wavelength here also. So with the help of this phase plate, the wavelength gets out of phase. Even the wavelength gets out of phase when it touches the specimen by 1/4, but when it passes through the phase plate, the wavelength gets half, 1/2 wavelength gets changed, or it becomes out of the phase. Those which passes through the undifractedly, they will not change their wavelength, they will remain as such. So this is the phase contrast microscope. You have to understand this point only, that there is a change in the wavelength. Then next we have fluorescence microscopy. We have to do four types of microscope: bright field, dark field. Then we have phase contrast microscopy, and the last is fluorescence microscopy. So what happens in the fluorescence microscopy? It exposes specimen to UV light, violet or blue light. Specimen usually stained with fluorochromes. Like in case of flow cytometry, where we are using the fluorochrome. Similarly here, the specimens are usually stained with fluorochromes, shows a bright image of the object resulting from the fluorescent light emitted by the specimen. So what will happen? We will get a bright image. But from which thing? From the fluorescent light we will get the bright image which we are able to see. So we are using fluorochromes here. Certain dyes such as fluorochromes, after absorbing UV rays, raise a higher energy level. So we are able to get the bright light. When the dye molecules return to their normal state, they release excess energy in the form of visible light known as fluorescence or the fluorochromes. So this is very simple. There is nothing in problem. If you are able to see, we have specimen here and this is the object. Now what will happen? We have different types of, you can say, mirrors or the detectors like in flow cytometry. So we are able to see the image which is quietly brighter in color. Like here you can see the background is darker in color. The background is darker, but the image which we get, they are quite bright. Why? Because of the presence of fluorochromes on it. So these are the, dyes which are basically used, like uramine, rhodamine, then acridine orange, which gives orange red fluorescence with RNA and yellow green fluorescence with DNA. So with the help of these dyes we are able to recognize the particular specimen, but here we are using the dye. In case of phase contrast, we are not using any type of dye. The phase contrast microscopy is based on the wavelength of the diffracted rays. Here we are using the fluorochromes. So if you can see here also, this is immunofluorescence in which we are using antibodies, and the antibodies will bind specifically to the dyes, and then this bacteria will give us the fluorescent color. So you can see the beautiful color of the bacteria. So what happened? We have a specific antibodies against the bacteria, but instead of binding to the bacteria directly, what we have done, we have dye and antibody interaction. So antibody binds with the fluorescent dye and we will get the color. So these types of fluorophores like DAPI, then we have fluorescein, isothiocyanate, we have cyanine, we have another types of, you can say, the dyes which are used to get a specific color. So this is the light source. Here the light source is on this way because this is fluorescence microscopy. So you can see the brighter color specimen, or you can see the fluorescent specimen. By passing through all these detectors or all these lenses, you will be able to see the bright color specimen. So if you can see here the comparison between everyone, this is the bright field. I am unable to see because of the okay. So this is the bright field. Here this is a bright field in which the background is bright and the specimens are little bit darker in color. In dark field the background is black and the specimens are lighter in color. This is phase contrast and this is the fluorescence, where the background is black but you are able to see the with the fluorescent dye you can see the specimen. So this is the difference between the four types of microscope, you can say. Even we have performed it on a single cell or a single organism, which is tetrahymena. When we see it under different microscope, the images are totally different. But in bright field and phase contrast, the images are little bit same, but here the images are totally different. So this is again a simple microscope. Let me show you one video. I am unable to see. So this is a basic. Yes. So here we will see the comparison.
Introduce you to the microscopy experimental technique and a basic light microscope. Microscopy is used to study the shape, morphology, and properties of cells, tissues, or organisms that cannot otherwise be seen by eye. Microscopy is also used to determine the presence or absence of certain types of cells or organelles in a sample or specimen of tissue, as well as to count the number of objects in a specific volume, for example, the number of cells in a sample. In this animation, we will provide you with an overview of optical or light microscopy, in which light is focused through a lens or a series of lenses to magnify or enlarge a sample. There are several different types of light microscopy, including bright field, dark field, phase contrast, confocal, and fluorescence. In this animation, we will touch upon bright field and phase contrast microscopy, and then we will discuss fluorescence microscopy in the next animation. All light microscopes have several components, including a light source such as a halogen light bulb or laser, a light source on-off switch with a knob to adjust its intensity, a diaphragm and condenser to focus the light onto the sample, a stage to hold the sample, controls to move the stage, focus knobs for coarse and fine focus adjustment, objectives to magnify the sample, and an eyepiece through which the researcher can view the sample. In addition, microscopes are often fitted with a digital camera and a computer for a scientist to capture images viewed through the microscope. To view a sample under the microscope, the researcher first places the object on the stage and then manipulates the controls, including the light, focus, stage position, and objectives, while viewing the sample through the eyepiece until a clear image is obtained. In this case, when the researcher is using a 4x objective, the image is 4 times larger than when it's normally viewed at this distance. When the objective is changed to a 20x objective, the image is now 20 times larger than when it's normally viewed at this distance. In a standard light microscope, the sample is magnified or appears larger by transmitting and focusing the light from the light source onto the sample on the stage using the condenser. The image of the sample is then magnified through a series of lenses in the objective and can then be viewed by the eye through the eyepiece. The amount of magnification that can be achieved by a light microscope is limited to the wavelength of light. As a result, objectives higher than 100x are not used on a light microscope, as it will have reached the limitations of its resolution. If a higher magnification is desired, then other microscopy techniques, such as electron microscopy, are used. A microscope is used to view many different types of samples, including cells growing in tissue culture, tissue samples, or whole organisms. While cells growing in tissue culture can often be simply placed upon the microscope, other samples need to be prepared and processed prior to viewing under the microscope. Many samples that a researcher would like to examine under the microscope are transparent and lack sufficient contrast to view a sample. Phase contrast microscopy can be used to view such transparent samples, such as unstained cells growing in tissue culture, as it increases the contrast of a sample. However, phase contrast microscopy cannot be used to view specific proteins or some cellular structures. To view, quantify, and localize particular cellular structures and proteins, a researcher uses dyes, stains, or antibody labeling. In histology, or the microscopic study of tissues and cells, different stains are used to highlight specific cellular structures, including hematoxylin, which stains nucleic acid structures blue, eosin, which stains basic structures such as a cell's cytoplasm pink, and oil red O, which stains lipids red, just to name a few. Expression, modification, and/or localization of specific proteins can be visualized using immunohistochemistry, or IHC. IHC utilizes a series of steps to first specifically recognize and bind a protein of interest using antibodies, and then convert this detection into a colorimetric signal that can be viewed under the microscope. We have now covered basic light microscopy and its primary laboratory applications. We will discuss fluorescence microscopy, which is a specialized type of light microscopy in the fluorescence microscopy animation. In this animation, we will introduce you to fluorescence microscopy. Fluorescence microscopy is a special type of light microscopy in which the microscope uses a laser as the light source. A researcher will require the use of a fluorescent microscope to visualize autofluorescent cellular structures, fluorescent dyes or stains, or to analyze the expression, modification, and/or localization of proteins using fluorescent antibodies by a technique called immunofluorescence. In a fluorescence microscope, the light source travels in a different trajectory than in the basic light microscope that we illustrated in the microscopy experimental technique animation. Instead of the laser light being transmitted through the sample, as in a simple bright field-based light microscope, the fluorescence microscope uses incident or reflected light. From the laser light source, the excitation laser beam travels through the filter cube, which is used to provide the appropriate wavelength of light to excite the fluorescent molecules, or fluorophores, used in the sample, then through the objective to focus the laser light on the sample. The light emitted from the sample is then reflected back through the objective and the filter cube, which filters the emitted light to the camera or eyepiece where the researcher can view the fluorescent signal. One advantage to fluorescence microscopy is that it can be used to detect and visualize multiple fluorescent molecules by simply adjusting the excitation and emission filters to detect the relevant fluorophores in a sample. Fluorescent microscopy also has many useful applications in the laboratory setting. Fluorescent dyes or stains are used to detect cellular structures. One example of a commonly used fluorescent dye is DAPI, which binds to DNA, and a second example is phalloidin, which binds to F-actin. Expression, modification, and/or localization of specific proteins can also be visualized using immunofluorescence, or IF. Like immunohistochemistry, IF uses multiple steps to detect specific proteins using antibodies in the sample. In IF, one of the antibody labeling steps utilizes fluorescently tagged antibodies that are detectable upon excitation using the appropriate wavelength of light.
So this is basically the fluorescent microscopy. I hope you will get it. Are you able to understand? So much saman dikhai. So are you getting my point, students? The type of microscopy which we have done. Is it buffering now also? So you are not able to see the video? Are you able to see the video? Let me change my direction. I am not able to see your answer. 1 minute. 1 minute. Okay. Yes. So are you able to understand what we have done? Hello. We are able to see, but it's a lot of buffering. We were able to see. Okay. So it's only a simple because of buffering. It's only a simple technique of phase contrast microscopy. If you are not see it, then I will show you it again. Kindly reply me. Are you able to see the PPT? आप लोगों ने PPT देख ली थी ना? It's only video plays on at that time. There is a buffering. Ma'am, we can't see you. Yes, there is a sun just behind me. I'm also feeling so uncomfortable. I'm unable to see your faces also. I'm just put it on another direction. Okay, let me put it on another direction. Wait please, wait please. Okay, I should. So I just changed my direction. I hope you will get me now. So is it okay now? I just changed my direction. Are you able to see me now? Gayatri, Debita, are you able to see me now? Or kindly reply, are you able to? Okay. So shall we move further or we will take a break of 5 or 10 minutes? Are you able to see the video of fluorescent microscopy, or I will show you once again when we will meet in within 5 or 10 minutes? Are you able to get the video? Please let me know. Now yes, I just changed my direction from first there is a sun just before me, so I moved into this side. So should I have to play the video again or is it okay? Because we have to do PCR today, Eliza today, we have lots of things to. Okay, you saw the video. Okay, very good, very good. One student is, I think, Devashish, she is unable to see the video. Then Devashish, I will send you the video. Need not to worry, we will share it once again also. So shall we take a break of 5 or 10 minutes, then we will start the next topic? Kindly let me know. It's 4 p.m. now, we have 1 hour more. So okay. So it's 4 p.m. now, 1 hour more. So shall we take a break of 5 minutes or not? Okay, then we will take a break of 5-10 minutes. Then we will start the basic recombinant DNA techniques which you all know. So we will take a break for 10 minutes. Is okay for all of you? Kindly let me know so that we will join at that time. 10 minutes is okay for all of you? Or 10 to 15 or 5 to 10, kindly let me know. So it's 4 p.m. We will meet at 10 p.m. Okay, very good. So it's 4 p.m. now. We will meet within 10 minutes. Okay. So you have to drink water or any type of things. So kindly do it. Then we will meet with recombinant DNA technology, PCR and ELISA. Okay. So should I end stream it now? I will share the link again when I will get live again. Okay. Should I end stream now? I will send the link again. Or keep it continue as such. Kindly let me know. Okay. So we have to keep it continue as such. Okay. So it's opening here. Okay. It's opening here. We will come back within 5 to 10 minutes and then we will start again. Is it comfortable to all of you? Yes, ma'am, and the stream. So it's like as such, you just join when you will come. Okay. So we will meet within 5 to 10 minutes. Let's take some break, then we will meet again. Okay. So take the break, we will meet. Okay.